Hello everyone, Assalamu alaikum. This is me Faraz Qurban Rajpar, and in this video lecture I am going to discuss about the structure activity relationship of benzodiazepine. We have already discussed the chemical structure of different benzodiazepine, and in this video lecture we will check out the effect of different functional group which can be substituted at the different site in the chemical structure of benzodiazepine, and we will observe its effect on the biological action of benzodiazepine. Structure activity relationship of benzodiazepine. To understand the relationship among the chemical structure and the biological action of benzodiazepine, first of all, it is necessary to understand the chemical structure of benzodiazepine. We have already discussed the chemical structure of different benzodiazepine in our previous video lecture. So over here we will take a general overview of the chemical structure of benzodiazepine. Benzodiazepine ring nucleus is basically consisting of the two main rings. One is the benzene ring, that is the aromatic ring, and it is fused with the heterocyclic 1,4-diazepine ring. This one is the benzene ring and it is fused with the this one 1,4-diazepine ring. The 1 and 4 position are representing the position for the nitrogen heteroatom that is present in the diazepine ring. Diaza represents the presence of the two nitrogen atom in this ring. The numbering of benzodiazepine ring is very much important to understand the structure activity relationship of different benzodiazepine. The numbering of ring will be started from this nitrogen atom, because this nitrogen atom is nearer to the ring of the benzene with which this diazepine ring is fused. So the numbering will start from here. This is the 1, 2, 3, 4, 5, 6, 7, 8 and 9. So these are the different position of the benzodiazepine ring. In this chemical structure I have represented the different substitution site, like this one is the R1, R2, R3, R7. So these are the different sites at which different functional group can be placed. Means the ring can be substituted at these sites, and each functional group will have a different effect on the biological activity of the benzodiazepine. The main biological activity of benzodiazepine is it acts as a CNS depressant. It acts on the GABA receptors. Its main therapeutic uses are it acts as an anti-anxiety, sedative and hypnotic depending on the class of benzodiazepine. So the functional group which are the other substituent like alkyl side chain or hydroxyl group and several other functional group which are replaced at these sites will have the different effect on the overall biological activity of the benzodiazepine. Some will have effect on its pharmacokinetic, like it may increases or decreases its metabolism and absorption, and some have effect on its half-life and so on. So now let's check out the effect of different functional group at each site. Aromatic and heteroaromatic ring of benzene. This benzene ring and this one diazepine ring is important or essential for the biological activity of benzodiazepine, because to produce its therapeutic effect, benzene must have to interact with the GABA receptor, and for its interaction with GABA receptor, the heteroaromatic and aromatic portion of ring is involved. This benzene ring will causes the pi-pi interaction with the amino acid residue of the GABA receptor, and this will causes the strong binding with the receptor, and due to which benzodiazepine can produce its biological activity. N1 substitution. Now let's check out that which type of functional group can be substituted at this position, N1 position. Most of benzodiazepine are usually unsubstituted, but substitution with the methyl group, that is the very lower alkyl, and with the halo alkyl and the amino alkyl, can enhances or increases its activity. So this one is the structure of diazepam, and in the diazepam it has the methyl group at the N1 position. So due to this N-methyl group, its activity is enhanced. And beside this, some benzodiazepine like halozepam has an haloalkyl, and some have the amino alkyl and other small chain alkyl group at this N1 site, and this is responsible for increasing its activity. Some benzodiazepine are unsubstituted at the N1 position, means they do not have any functional group or any alkyl side chain at this position. Example of those amino acids are oxazepam, lorazepam, nitrazepam, and others. C2 position. The presence of carbonyl or ketone group at the second number position of benzodiazepine is essential for the benzodiazepine activity. We have already learned the structure of different benzodiazepine, and we have observed that almost all benzodiazepine contain the ketone group or the carbonyl functional group at the carbon number 2 position. So if we are going to reduce this carbonyl functional group and convert it into the methyl or CH2 group, this will causes decrease in the activity of benzodiazepine, and due to which its potency of the drug will also decreases. Some benzodiazepine lacks the carbonyl or this ketone group at carbon number 2 position, but they have certain other functional group which are responsible for their activity. Like chlordiazepam has the sulfonyl functional group, and the chlorodiazepoxide has the methyl amino group. But these type of benzodiazepine also require bioactivation inside after the metabolism. They are going to be converted into this carbonyl functional group. Like chlordiazepam, sulfonyl group undergo through the oxidation, and it will then converted into this carbonyl functional group. So the most important functional group at the carbon number 2 position is the carbonyl functional group, that is responsible for producing the therapeutic action of benzodiazepine. C3 substitution. This carbon number 3 position of benzodiazepine is also responsible for the benzodiazepine biological action. If it is being substituted with the alkyl group like methyl, ethyl, or propyl, or any other alkyl side chain, this will causes the reduction in the biological activity of benzodiazepine. So means for the biological activity of benzodiazepine, it must not be substituted with any type of alkyl group at the carbon number 3 position. But there is an other polar functional group, that is the hydroxyl functional group. Hydroxyl functional group presence or absence can be essential for the benzodiazepine activity. There are certain type of benzodiazepine which do not have 3 hydroxyl functional group like diazepam, nitrazepam, clonazepam, and many others, but they have the essential biological activity. Due to lack of 3 hydroxy functional group, as in this structure of nitrazepam, you may observe that there is no any hydroxyl group. So due to this, it is a non-polar in nature, and these kind of benzodiazepine are basically the long acting. They have the longer duration of action and long half-life, because they first have to undergo through the phase 1 oxidation reaction or hydroxylation at this site, which will impart or introduces the hydroxyl functional group, which enhances its polarity, and then they will undergo through the phase 2 conjugation with the glucuronic acid. So the most important functional group that can be substituted at the C3 position is the hydroxyl functional group. So there are certain drugs which have the hydroxyl group at the C3 position, like oxazepam, lorazepam and temazepam. And these drugs, like this is the structure of oxazepam. Oxazepam has the hydroxyl functional group at the carbon number 3 position. And due to the presence of this hydroxyl functional group, this drug is polar in comparison to those which lack the hydroxyl functional group. And it has the shorter half-life and shorter duration of action. And it is a short-acting or intermediate-acting benzodiazepine, because it can directly undergo through the phase 2 conjugation with glucuronic acid. Glucuronic acid will form the glucuronide at this 3-hydroxy substitution site. We have already discussed the reaction mechanism of metabolism of different benzodiazepine in our other metabolism of benzodiazepine lecture. So basically, if the drug have the 3-hydroxyl group at the carbon number 3 position, it will have effect on its pharmacokinetic. How? It will causes the rapid metabolism of drug. It will directly cause the phase 2 conjugation, which will causes the reduction in half-life of the drug and shorter duration of action of drug. Unsaturation. Unsaturation, or the presence of double bond, is also essential for the biological activity of benzodiazepine. But the position of unsaturation in the chemical structure of benzodiazepine is at the carbon number 4 and 5, at this position. So the carbon number 4 and 5, the unsaturation must be present at this position. If saturation of this double bond occur, this will cause the reduction in biological activity of benzodiazepine. Or if we shift this double bond from 4-5 position to 3-4 or any other position in this diazepine ring, this will also decreases the biological activity of benzodiazepine. So the unsaturation or double bond must be present between the 4 and 5 carbon of the benzodiazepine ring. C5 phenyl ring. The C5 position of the benzodiazepine at the diazepine ring must be substituted with the phenyl ring. The phenyl ring is essential for the biological activity of the benzodiazepine. Further, we can also substitute this phenyl ring with certain type of other functional group, like electron attracting group, which are the electronegative group, by substituting it at the ortho position, like with the chlorine group. Halogenatom group can be substituted at the ortho position. This will further increases the potency and increases the activity of benzodiazepine. But if this phenyl ring is substituted at the meta position or para position, this will causes the reduction in the biological activity of benzodiazepine. Like in this structure, in this chemical structure, the benzodiazepine is being substituted with the second position of the phenyl ring with the chlorine that is the electronegative or electron attracting group. C7 position. The carbon number 7 position of the benzodiazepine. This one must be substituted with the electron attracting functional group like halogen atom, chlorine, fluorine, bromine or nitro group. This will increases the biological activity of benzodiazepine, and this has also effect on the potency of benzodiazepine. If the benzodiazepine have the chlorine, fluorine or nitro group at this carbon number 7 position, it will have high potency, means it will increases the activity of benzodiazepine. So at C7 position there must be presence of any electron attracting group like chlorine, fluorine and nitro. The position 6, 8 and 9 of the benzodiazepine ring must be unsubstituted. Why? Because if any type of functional group is substituted at this position at 6, 8 and 9, whether it is any alkyl side chain or any other particular halogen or any other functional group, it will result in the loss of benzodiazepine therapeutic activity. Means if any benzodiazepine have any type of substitution at these three sites, then it will ultimately have no any biological activity due to these functional group. So for the essential activity of benzodiazepine, for its optimum activity, these three sites in its chemical structure, that is position number 6, 8 and 9, must be unsubstituted. Triazolo benzodiazepine derivative. The triazole is an heteroaromatic ring that can also be substituted at the carbon number 1, 2 position of benzodiazepine. This will produce certain type of new derivative of benzodiazepine that are highly potent, that will have the high potency. Like examples are alprazolam, triazolam and acetazolam. These benzodiazepine are high potent drug and they usually have the shorter duration of action, and they are rapidly metabolized by alpha hydroxylation of the methyl group that is present at the triazole ring site. This triazole ring is fused with the diazepine ring at the 1 and 2 site by the hydrogen bond interaction. Imidazole ring fusion. Benzodiazepine derivative can also be synthesized by the fusion of imidazole heteroaromatic ring at the carbon number 1, 2 position. This will synthesize or produce certain derivative, that is the midazolam, that is also ultra short acting benzodiazepine. It also has the shorter duration of action due to the rapid metabolism. Now let's check out the summary of SAR of benzodiazepine. In this video lecture, we have observed the effect of different functional group and the heterocyclic ring at the various sites in the chemical structure of benzodiazepine, and we have also observed its relationship with the biological activity of benzodiazepine. Like if 1 and 2 site of benzodiazepine is being substituted with the triazole or imidazole ring, it will result in the highly potent benzodiazepine like alprazolam, triazolam and acetazolam. If N1 site of benzodiazepine is replaced with alkyl, haloalkyl or amino group, it will result in the increase in the biological activity of benzodiazepine. But some benzodiazepine lack the alkyl or any other functional group at the N1 site. The carbon number 2 position of benzodiazepine must have the carbonyl functional group or ketone group. This is the essential for the benzodiazepine. We have already discussed the chemical structure of various benzodiazepine in our previous video lecture, and we have observed that carbonyl group is almost present in every benzodiazepine at carbon number 2 position. But some benzodiazepine like clozapam have the sulfonyl functional group, and it went to the oxidation reaction of metabolism and converted into the carbonyl. The next is the presence of the unsaturation. Unsaturation must be present at the between carbon number 4 and 5. This is also essential for the biological action, that is the CNS depressant activity of benzodiazepine. If this unsaturation or double bond is being replaced with the single bond, or it is saturated, or it is being moved to another position, then it will result in the loss of biological activity of benzodiazepine. Phenyl group must be present at the carbon number 5 position, like this is the phenyl ring. This phenyl ring is also essential for the benzodiazepine chemical structure and its biological activity. If benzodiazepine lacks this phenyl ring at C5 position, then it cannot produce its CNS depressant activity. C7 position of benzodiazepine must be substituted with the halogen or any other electronegative atom like nitro group. The presence of this will result in the highly potent benzodiazepine. The 3 position of benzodiazepine must be substituted with or without hydroxyl group. There are certain benzodiazepine which have the hydroxyl group and some do not have the hydroxyl group. Hydroxyl group will have the effect on the metabolism or pharmacokinetic parameter of benzodiazepine. But this C3 position must not be substituted with any type of alkyl side chain like methyl group. If methyl group is present at the carbon number 3 position, it will result in the loss of biological activity of benzodiazepine. C6, 8 and 9 position of benzodiazepine chemical structure must be unsubstituted. If these sites are substituted with any type of functional group, it will result in complete loss of biological activity of benzodiazepine. So this is all about the structure and its relationship with the biological activity of benzodiazepine. Thank you so much everyone.